Take it away, Carrie.
All right, thanks, Jean, and thanks everyone for being here. I have the pleasure to announce Tracy Thomson. Tracy is a Utah native who completed an associate's degree at the College of Eastern Utah and a bachelor's degree at the University of Utah. He holds a master's degree from the University of California, Riverside, and just graduated this past summer with a PhD degree from UC Davis. His academic interests include paleoecology, ichnology, which is trace fossils, vertebrate paleontology, and functional morphology. He is currently working as an adjunct professor for Woodland Community College and a research assistant at UC Davis. And most importantly, he worked at the NHMU with me for a few years, and so that was really important too. So we're very happy to have Tracy here. So thank you so much, Tracy, and take it away.
Okay, thank you, Carrie. I'll get my presentation shared here. Okay, so I think that should be working now. So what I'm going to talk about tonight is kind of encapsulates my PhD work at UC Davis about the claw function and with an application to Archaeopteryx and trying to interpret claw function in fossil organisms, specifically in Archaeopteryx, kind of as a case study. And so kind of a roadmap for what I'm going to be talking about tonight is we'll do a little bit of a setup to set up why this is important and kind of the problems that I'm trying to tackle here. There's kind of three parts that correspond to the three chapters of my dissertation, dealing with the claw functional morphology itself and kind of setting up the methodology for attempting these things. And then I did a little bit of study and insights into the claw structures themselves, specifically the ungual sheath relationships. So claws are composite structures. They have a bony ungual on the inside covered with a horny sheath on the outside. And I wanted to kind of look at the relationships between those two structures, particularly with fossil organisms where a lot of times the sheaths don't preserve. And so we just only have the bony ungual, and I wanted to look at those relationships. And then part three was applying all of this information and whatever I learned to interpreting claw function and also ecology and evolution in Archaeopteryx. And then I'll kind of arm wave a little bit about some future work that I may or may not get to at some point in the future, but things that I would like to work on related to this topic. So a little bit of the setup, specifically with Archaeopteryx. Archaeopteryx is widely regarded as one of the first birds, definitely one of the first aviolans, and it has a really close relationship to the Deinonychosaurian dinosaurs, so things like Utahraptor, Troodon, and Velociraptor. But it also is related to this other group that I want to point out, the Scansoriopterigidae, and one of those species being Ambopteryx. And these are a really weird group of dinosaurs that evolved close to the origin of birds, and we'll come back and talk about them a little bit later as well. But this is the phylogenetic framework and the family tree, as it were. And so Archaeopteryx and some of these other dinosaurs as well are really important for trying to understand the evolution of flight in birds in this group. They have these interesting transitional features, and we want to try to understand the ecological context in which they evolved powered flight. And a lot of the arguments around how flight evolved kind of revolve around this idea of, was Archaeopteryx or these proto-birds, were they predominantly arboreal, climbing and perching and gliding through the trees, or were they predominantly cursorial, running along the ground, catching prey, and that's where flight developed. So again, the ecological context of the evolution of flight in this group, Archaeopteryx is a key species for trying to understand and interpret that process. So how do claws help us in that? Well, it's long been known that claws give some indication of the ecology of the organism. As far back as 1993, Fiducia looked at Archaeopteryx in comparison with other birds, and they measured the claw curvature using angles in this method that I also use up here. And they showed that when you go from ground dwelling to perching birds to climbing birds, the claw curvature increases as you go through those groups. And then he looked at the claw curvature in Archaeopteryx, and he determined that Archaeopteryx claws fall within the perching and climbing threshold of the claw curvature. However, some some workers after him came along, and they've determined that the claw curvature actually shows that it was a ground-dwelling bird in some cases. Down in the bottom left is one of those. Or in other cases, like in the bottom right, the Burn-Jeffrey et al., they determined that Archaeopteryx is kind of a middle ground generalist type. And this is all based on the claw curvature and other aspects of the claw. So we're having a problem here where we're not agreeing on what the claw curvature actually means. And I think that's due to a couple of problems that I try to solve in my PhD work and that I'll go into here in a second. So the way I saw it is that there were three major hurdles to being able to confidently infer claw function and ecology in fossil organisms. The first was the morphological metrics themselves, how you're actually measuring the claw. Predominantly, claw curvature has been the only metric or only one of maybe three different metrics used to try to interpret the ecology of the animal. And that's too little. There are far more aspects of the claw. There's cross-sectional shape, there's the length and the width and the height and the flexor tubercle size, all of these aspects that aren't taken into account. So I think we're missing a lot of the functional ecological information in the claw by only focusing on the curvature. So I wanted to add more measurements to see if we could clear that up. The second hurdle I saw was that the categories themselves. We saw that categorizing animals as predatory, climbing, perching may itself be too simplistic. And so I would propose that we categorize animals not based on life habit or even ecology, but on the function of the claw itself. And this requires actively looking at the claw function and interpreting that function specifically and distinctly. As an example, you can see on the bottom right down here, I have a column of predatory animals, Komodo dragon, a wolf, Siberian tiger, and a golden eagle. These are all predatory animals. You would put them in a predatory category. However, if you look at their claws, they all look different. All the claws look different. So you're blending and mixing into this category just by putting them into that category. And then conversely, if you look at the bottom row, we have a golden eagle, woodpecker, and a robin. This is a predatory bird, a climbing bird, and a perching bird, and the claws look very similar. So having a sense of what the function of the claw actually is, and not just lumping everything as predatory or as perching, needs to happen. And then specific to fossils themselves, I mentioned this before, is the soft tissue preservation, or I call it the soft tissue issue. This is where the bony ungule that is inside the keratin sheath, for the most part in fossils, the keratin sheath, the soft tissue decays and isn't preserved. So here's a golden eagle claw, and if I place the sheath over the claw, that's what the sheath looks like on the claw. So that's the whole claw. So if we're looking at fossil organisms and we don't have the sheaths to reference or to measure, how can we trust or have confidence that what we measure on the ungule actively represents what the sheath looks like, and then stepping off from that to infer function and what all of this means? So these are the three major hurdles that I tried to overcome in my project as far as getting to this step where we can actually interpret function in fossil claws. So related to those, the three major goals where I wanted to develop a methodological process that would inform the best practice for going about this problem. How do we predict claw function in fossil specimens, and what is the best way to do that? I also wanted to describe the morphological relationships between the keratin sheath and the bony ungule, specifically because, again, with fossil vertebrates, sometimes we have the bone but not the sheath, and I wanted to look at those relationships. And then take everything that I learned from that and apply it to a case study. And in this case, we chose Archaeopteryx. So in the third part, I will apply all of this to Archaeopteryx, and we'll see what happens with Archaeopteryx. So for the project, I had to go to several museums to look at modern claw specimens of modern organisms. These are various museums in California, the Cal Academy, Museum of Vertebrate Zoology of Berkeley, and the LA County Museum. Go into their drawers, go into these boxes, and in the center photo, you can see a box of two big boxes here, and they're labeled Struthiocamelus. So that's an ostrich. So these are boxes of ostrich bones. And I had to sift through these bone boxes, pull out the claws, and use those to measure for my analysis. Now, museum specimens usually come in two flavors. There are skins, which you can see on the left. These are basically like taxidermied specimens. They have all of the soft tissue intact, at least the skin and the claws and the sheaths and the beaks and everything like that. And then on the right, you have skeletonized specimens, which is just the skeletons left behind. Now, one of the issues with using the soft tissue or the skin specimens is that you can't see the proximal end of the claw where it articulates with the toe. The flexor tubercle and the bony parts of that are covered with muscle and tissue, and so you can't measure those parts, whereas you can measure those in the skeletonized specimens. However, the skeletonized specimens, often the keratin sheath doesn't preserve because it's soft tissue. So in the skeletonized process, a lot of these specimens don't have sheaths. So I went back and forth on what's the best way to use these specimens. I looked at the soft tissue ones first, the skin specimens. And on the right, there were several methods that I could have used to image these things, to look at them in detail and measure them. You can use X-ray, CT scan, or micro CT scanning. We could even use photogrammetry to look at the three-dimensional structure as well. But these all had problems. One is that the X-rays didn't have the enough resolution to actually see the very small claws on things like falcons and small woodpeckers. The micro CT scan would be able to pick up that detail, but micro CT scanning, it's super expensive. And if you have 80 specimens, which is what I ended up with, that goes into the several thousands of dollars to do that. Photogrammetry is good, except that it's difficult to properly scale things of different sizes. When you're trying to make sure your measurements are accurate across a wide variety of shapes and sizes, photogrammetry was going to be a little bit of a difficult process for me. So I decided to go completely old school. I went with the skeletonized specimens that did have sheaths. Sometimes the sheaths were stuck to the bone, and so I had to boil the claws to soften them up so that I could separate them out. Initially, that boiling took place in my kitchen, in my pan. But then eventually I moved into the lab and I got myself a normal hot plate and everything that I could boil these things in and separate the sheaths out. So that's how I went about that process. And then once I had them separated, I could actually make molds of the bone and the sheath separately. I could pour resin casts of those and then cut the casts in cross section, scan the cross sections using a scanner, and then I would have a digital image of the cross section that I could run through my computer program to make the measurements. So that's how I did that process. For looking at the claws in side view, I would take a picture of the claw with the claw and the ungual together. That's in the left-hand side. And then I would take the sheath off and take another picture of the claw separated, so the sheath and the ungual next to each other. And then using Photoshop and various techniques, I could actually cut those out, and then I could overlap them back together using the original photo as a reference. I can overlap these back onto each other to produce what the claw looks like in life. And then because they're two separate images, I can play with the contrast and the transparency and the colors to be able to look inside and see what the ungual looks like. And then make measurements of the ungual in life position and its relationship with the sheath measurements as well. So that's what you can see there. And because I can play with the color, I was able to produce some really nice false color images of some of these claws. So you can see the ungual almost like the sheath is translucent. And this made it not only really cool to look at, but also very easy to make measurements on these structures. Now to make the measurements themselves, oh yeah, here's these are all the specimens of most of them that I came up with that I used in my study of all the various animals. Okay, so let's talk about claw function then. So I mentioned that I wanted to come up with a lot of different parameters and a new classification based on function rather than ecology. I want to show this video of a peregrine falcon consuming a mouse. If you're a little bit squeamish, you might want to look away, but it's not that bad. Let me just get that started here. So if you look, after the peregrine falcon has caught the prey, it uses its claws to wrap around the prey and squeeze and suffocate the prey, but then it uses the inner digits of the toes to pin the prey to the ground while it picks off bits of flesh. And you can actually see it repositioning itself to actually get those digit 2 claws to pierce and hold down the mouse or the little rodent as it takes its bites. So this just demonstrates that claws can have multiple functions. There isn't just one thing that the claw does unless it's a very highly specialized animal. But you really need to pay attention to what the animals are doing with their claws and use that to classify the claws and the species. So these are the categories that I used in my study. This is based off of 330 different species and 390 references or scientific literature papers that support ascribing these animals or these claws to these functions. This is a database that I continue to grow and add to, but this is the database that informed the functional categories that I eventually assigned these claws to. And there's a list in there. I'm going to go through these one at a time just so we have an idea of what each of these functions is. The first one is Amplectorial or grasping. Birds of prey do this, that they'll do, I mentioned already, that they will wrap their digits around the prey animal and then squeeze it to death. There's no puncturing of the animal involved. If there is, it's accidental. It's not the primary mode that it uses to kill the animal. So these are birds that wrap their claws around an animal. This is the same function or same biomechanics that perching birds use to grab onto a branch. So it's not surprising that in my analysis, actually predatory birds and perching birds actually group together because the function is the same function. The claw function is the same function. I had cursorial animals, anything that is running or hopping over long distance in flat terrain, things like kangaroos, roadrunners, and ostrich. These are cursorial animals, and the claws basically function as hooves to make a nice flat surface contact with the substrate for friction, so they can run fast, they have a good push off of the foot, and they can turn and maneuver using that surface of the claw. There are griporial claws. This is a hook and pull style digging. So many armadillos and anteaters use it this way. What they'll do is they'll insert their claw into the substrate and then they use a rotational motion to loosen the substrate and then dig that out. So there's an insertion and a rotation motion involved with this function. This is different from scalporial, which is scratch digging. This is just a front and back motion. So you can think of like meerkats when they're digging in the soil. That's what they're doing. What's really interesting is I discovered a lot of species like the puffin that do scratch digging with their claws. Puffins will scratch dig out with a claw on their foot. They will scratch dig out their little nests that they lay their eggs in. So again, it just brings home the point that you really need to pay attention to what the animals are doing with the claws and not just assume that you know what the function is based on what they are or where they live. I have a Scansorial category. This is the climbing category. Things like tree kangaroos, squirrels, and downy woodpeckers. The squirrel and the woodpecker will be important later on. I'm going to talk about them more later when we look at Archaeopteryx in detail. And then there's a suspensorial category. This is only sloths. I haven't found anything else that uses its claws like sloths do. They will wrap the claw over the branch and then just hook it there and suspend themselves from that claw like this. That's different from grabbing the branch. It's a different function, different physics are involved, and so it's a unique category, but it's only one that's occupied by sloths so far. I haven't found anything else that does this. And then there's this really cool category that I call Tensasorial. This is the grappling category. Things like cheetah and osprey. These are animals that they actually will pierce the flesh or pierce the object with the claws, and then they act like Velcro, where they grip and grab hold of that object with this grappling motion. Really interesting. There is several species called fishing bats that will use this on their legs. They will swoop down over the ponds and they will run their claws through the ponds and they will grapple and grab up fish and insects off of the water surface and scoop them up into their mouths as they fly away. And they fish for things like that. So that's really cool. And then I did have a generalist category. There are some species like the caracar here and the crows that are very multi-purpose. It was very hard to put them into any one category. The caracars, for example, they will stand by stream beds and they will fish food out of the stream. They'll perch. They will grab animals and hold them. They're very smart birds as well. What's really interesting is I found instances where they would wait in trees by a bird nest, and when a truck would go by, the birds would get scared, leave the nest, and the caracara would swoop in and grab the prey out of the nest. It would lay that trap and wait for that scenario and then go in and grab it. There's also documentation of caracaras working in groups of 2 or 3, where one of them will overturn a stone and hold it while the others go in and grab food, and they'll take turns doing this, cooperating with each other to search for food and to obtain their food. Really smart birds, and something that I'd like to look into more detail is how their behavior operates. And then the morphological metrics themselves, I mentioned that I wanted to use a lot more, so I use about 32 of them, not just curvature, but all sorts of things. And the takeaway I wanted to show you here is with the curvatures. So the curvatures are measured in angles. The problem with using angles is that everything else is measured in lengths. And when you throw length measurements, in this case in the units of millimeters, into an analysis with measurements in other units, in this case degrees, you're mixing the units, and mixing the units in statistical analyses isn't a good thing. So what I did was I came up with a way to describe the angle in terms of lengths of the sides of the triangles that form that angle. So that's where the length and the radius comes in on some of these angles. And what I did was I analyzed the morphological metrics separately, one using the mixture of degrees and lengths, and one using only the lengths, just using the degrees expressed as lengths. And then the last thing that I used here is what's something called the second moment of area. That describes the cross-sectional shape. And what that is it's an engineering term. They use it to describe the strengths and the shapes of beams in construction. And so I used it to describe the shape of the cross-sectional area. That's called the second moment of area. I used a computer programming language. I wrote scripts in MATLAB and lots of different functions that talk to each other and do all this for me, so that I didn't have to do it by hand every single time. So I wrote all of these scripts. This is what the program looks like, where you import those photos that I mentioned earlier, the cross-sectional shape and the side view of the claws with the ungule and the sheath. And then you click the points using the computer mouse, and then the scripts work out all of the math for you and log all of the data as it were. So this was really helpful because sometimes I wanted to go back and change how I did things, and it was very easy for me to just click through and do this. So it took some time up front to write all these scripts and to write all this code, but on the back end it saved a lot of time because I could easily adjust and add new specimens without spending a whole afternoon measuring a specimen. It made it very quick and easy. Here's all the various analyses that I did. I'm not going to go into how all of these work or anything. The biplots and the box plots are pretty self-explanatory. You're just comparing two variables and looking at it, and we won't look at too many of those. The principal component analysis, that just looks at shape difference. What that does is it takes all the measurements of the claws, and the computer works out how you can best separate those measurements, or how to best represent those shape differences in two-dimensional space. I don't look at those so much because what ended up happening is that size became a huge factor in the PCA analysis, and I didn't want to look at size. I just wanted to look at shape, but the PCA doesn't know the difference. It looks at size as well as shape. So it wasn't really that helpful. What was really helpful was the linear discriminant analysis. What this does is the computer takes, again, all of those morphological metrics, looks at the functions that I've assigned to them, and draws up, uses kind of like machine learning to develop a set of equations and methods to then classify those specimens into their respective categories. So it takes all of this information and then asks the question, if I give it an unknown specimen, like for example Archaeopteryx or something else, how confident can I be that this program will classify that correctly into one of these functions? And it will give me numbers for all of these different species and everything. One of the really cool things with this analysis is that it also gives something called posterior probabilities. So nothing is 100% classified as scansorial. Maybe it's 90% chance that it is, or an 80% chance. And what I can do is I can look at those probabilities. It may be 80% one thing, 10% another thing, 5% another thing, right? It will break up those probabilities. So I can consider not just what it wants it to be, but what the second most likely thing it could be, or the third most, and so on and so forth. So that gets interesting to look at. And then lastly, the permutation and PFDA tests. I'm just going to mention the results of these. This checks for randomness in the data and phylogenetic bias. So what we did is there's the possibility that all of these measurements that I took and all of these functions that I described and the correlations between those measurements and that function are due to random chance, that there is no correlation, that there is no causation between the shape and the function. So we ran a permutation test, and it turns out that it is, the number was ridiculous. It was 0.0 times 10 to the negative 15th chance that it was random. Very, very, very low. I mean, essentially 0% chance that the correlations that I'm finding between function and shape are due to random chance. So that's not what that is. The other thing was the PFDA test. Now, some of these claws could appear similar to each other because they're similar animals. So all the birds could clump together because they're all birds, not because they're doing different things with their claws, but because they're all closely related. So the PFDA test tested for that bias, that there may be, it's called a phylogenetic bias, due to the evolutionary history and close relatedness to the specimens. So we did a check for that, and it turns out that there's very little, if any, phylogenetic bias in the data, which is unusual for these types of studies. Usually there is a significant amount of bias that you have to statistically correct for to be able to have confidence in your results. What we ended up seeing was that there was very little to no bias. I think that's a result of the number of morphological metrics, the number of measurements that I took kind of overwhelmed any sort of residual bias that there might have been in the results. So that's good news. So we can have good confidence with the studies, and let's dive into what we find. So no surprise, I found that as you go from digging animals to cursorials and generalists and on up to Amplectorial and Tensasorials, the claw curvature increased. This is what previous workers have found. It's just parsed out into more distinct categories, but the trend and that pattern follows. So there was no surprises there. What is interesting is if you look at the Tensasorial category, these are things like osprey, fishing bats, and cats. They have very, very high degrees of curvature on them, more than anything else. Everything else kind of overlaps with the other categories, but the Tensasorials really stand out when it comes to the curvature angle, at least the inside curvature angle of the claw. So that may be one way to tell a Tensasorial animal right off the bat, is look at the inner curvature, and if it's very high or in this range, you're probably dealing with a Tensasorial animal. I did look at this very interesting metric. This is a logarithmic spiral. So a lot of things in nature grow in spirals, and claws are no different. However, when you look at different animals with different claws, sometimes the spiral fits. If you look at the lynx rufus, that's a bobcat claw, you'll see the solid line on the outer edge fits very, very nicely to the claw. But the inner line that I tried to match to the ventral portion of the claw doesn't match very well the margin there. And the same thing is true in the dasypus, except in this case, both the dorsal margin and the ventral margin don't fit a logarithmic spiral very well. So what that meant for my analysis is that the logarithmic spiral, or trying to match a spiral to the claws and use that as a morphological metric, is inadvisable. It doesn't work out very well. If you look at the box plots on the left, you'll notice that all of the log spiral measurements that I made are all overlap with each other. There's no one group that stands out. There's lots of overlap in the measurements, and so it's not a useful metric. So I ended up eliminating it from the analysis. It wasn't helping in any way to distinguish function, and so I ended up not using it. Interestingly, though, you can use it on shoreline evolution. On the bottom picture there is Bodega Bay out there on the coast of California, and it has a spiral shape as you go from north to south. And with my computer program, what's been fun is I take these satellite images and I can click dots along the beach and match a spiral to the beach like I've done with the claws. And you can describe these beach lines in terms of logarithmic spirals. And so that's kind of been a fun thing to do. So I'm going to jump right over to the LDA, the linear discriminant analysis. Remember, this is classifying. So the more separated these groups are, the better the classification is. And what you'll notice here is that a lot of these groups, there is some overlap, but they are also separating quite a bit in this space. So you can see where there's some overlap. Some of the overlap is predictable. For example, the generalists in the green square, they're going right in the center of the LDA, and that's where you would expect them to be. Being a generalist is in the middle ground. And then you can see everything else around the edges there. I do want to put up the numbers. I mentioned the raw data set. The raw data set is the one that has the degrees, and the length data set is the one that has lengths. So remember, I separated the degrees from the lengths to see if that had any effect. In the raw data set, it was able to correctly classify 75% of the species. If you remove the second moment of area, if you remove the cross-sectional shape, it drops to 72%. So not a huge drop there by removing the cross-section. With the length data, it's significantly better. It's 84%, almost 85%. And again, removing the second moment makes a significant drop down to 76 or 77% correctly classified. So this is pretty good news. If we look at the posterior probabilities, this is looking the 75% for the raw data set correctly classified. That is looking at what the analysis wants it to be. It's first choice, right? It's highest probability. If you consider not just the highest probability, but also the second highest probability for each one of these, that classification rate goes up to 90%. So if you have some doubt over what the program is classifying, and you take a look at its second choice or the second most probable, then you get into the realm of 90% or better classification rates. The length data set was even better at 96% when you consider the second most probabilities of these things. An interesting phenomenon is, you look at the numbers there, is that mammals are being classified more often correctly than the birds. 71% of birds, 78 to 90% of the mammals. I figured that may be to two things, result of two things. One, maybe the data set contains more mammals, and so there's more information for it to use to make its predictions. Or the second option might be that claw morphology is more conservative in birds than in mammals. If birds have claws that are all more similarly shaped to each other, and mammals are more disparate in the claw shape, that's more information that the program can use to deal with the mammals, and less information that it can use on the birds. I tested both of those hypotheses using the PCA and LDA holes. I'm not going to go into how all that worked, but what it turned out to be is that the data set contains more mammals than birds, and that's what was causing it, the mammals to be classified correctly more often than the birds. This is a double-edged sword, good news and bad news. What it means is we need more specimens, but what it also means is by adding more specimens, the prediction will get better. So future research, adding more specimens, more species, and more functions in there is only going to make this process better. It's going to improve it. All right. So we learned that we have to pay close attention to morphology. We can't just rely on assume function. Look at the literature and make sure the functions are there. Also, the details matter. And using appropriate functional categories and morphological metrics does make a difference. We're actually able to parse these things out now and make accurate predictions, whereas before that wasn't working out. Also, representing angles in terms of lengths, not using degrees, but using length-only measurements should be the preferred method because it increases the classification rate. More things are classified more correctly. Okay. So I want to talk about a couple of things that I found with claw structure, because that becomes important when we're trying to infer function in fossils. These are just some of the things that I was testing. So for example, how reliable are the functional inferences based solely on the ungual versus sheath? The previous section, I only looked at the sheath. So if we only have unguals, if we have a fossil that doesn't preserve the sheaths, how does that work? Can that still be accurate? Is the ungual an accurate proxy for the sheath in those instances? What if we have both? If you have an animal like Microraptor, or there are some Archaeopteryx specimens, the ones that I use in particular, that have both the ungual and the sheath, is that important? How much better does the classification get by using both structures? And if the sheath morphology does correlate to ungual morphology, then we should be able to predict claw function in any fossil organism, regardless of whether we have the sheath or not, or whether we have the cross-sectional area or not. So if we keep in mind that a lot of these fossils, especially in Archaeopteryx, are squished or crushed, and so the cross-sectional area is not obtainable, even through CT scanning or 3D scanning, because the fossil has been squished. And so I wanted to investigate how we approach that scenario when we don't have the cross-section. So I used the same metrics. I just used all the ones that I did for the sheath, made the same metrics on the bones, and lumped them all together into one big data set that I could then look at separately, together, all of that. So here we go. One of the cool things that I found was that cats are unique in their claws. That's kind of intuitive, but here we have some interesting things. If you take it metric by metric, feature by feature, sometimes the sheath of the cat claw really matches the ungual portion. In the case of the cross section, the bottom right here, the second moment in the X direction, very tight correlation. The cats aren't sticking out. They aren't separating themselves from the rest of the animals very nicely there. Whereas, for example, in the upper left, the ventral curvature angle, the ungual does not accurately reflect the sheath shape at all when it comes to cats. And then there are cases in between in the other two figures here. So cats, what I ended up doing was removing cats from the analysis and putting them back in to see how that affected the results. And again, it just turns out that cats are really unique. I'm not going to go through all of these numbers, but if you look at the R-squared numbers in both of these columns with the cats included and cats excluded, that R-squared number, the closer it is to 1, the closer the relationship between the ungual and the sheath for that metric. The first metric on the list, for example, is the ventral curvature angle. With cats included, because it's such a horrible correlation in cats, the R-squared number is very low, 0.21. But when you take cats out of the equation, being very specialized, that improves to 0.6. Still not great, but a significant improvement. Looking down at the bottom, you can see the second moment of area metrics, the cross-sectional shape metrics. Doesn't matter whether cats are included or excluded, and a very tight relationship, 0.94, 0.96, very close to 1 on those relationships. The other interesting thing that I found is that these relationships are nearly all isometric or slightly negatively allometric. What that means is that it doesn't matter if you're looking at a small claw or a big claw, the relationships hold. Or they're slightly negative. What that means then is that as you look at a big claw from a big animal, the ungual has to, in the case of curvature, for example, the ungual has to take up more of that curvature to support the sheath, whereas in a smaller animal, less of the curvature is taken up by the ungual. I suspect that in bigger animals, the ungual has to take up more of the stress of the function of that animal, and so there's a slight negative allometry to it. But not so much that that size becomes an issue. Again, whether we're looking at big claws or small claws, the relationships hold across all of these metrics, which is pretty exciting. These are just PCAs of the shape disparity in unguals and sheaths. And what this shows is that they're not completely overlapping. What that means is that there is shape information that the ungual has that the sheath doesn't have, and vice versa. So using both of them together, you get a complete picture. Using one by itself, you miss information in the other, and vice versa. So even though they overlap quite a bit, there is some space that is not shared between the two structures, and that means that using both structures will give a better result. And that's shown here. These are all of the options that I went through. Do you have just the sheath, just the ungual, or both together? Are you using the raw data set versus lengths? Do you include the shape, the cross-sectional shape, or not? I haven't mentioned the extreme morphology. There are some claws of some animals that are just really out there weird and unique, and when you add them into the data set, they bias the data and it doesn't turn out well. So what I figured out that if you only have the sheath of the animal, use the lengths, include the second moment, the cross-sectional area, and take out any unusual, really weird stuff, you can get the misclassification rate down to 15%. That means about 85% are classified correctly. If you only have the ungual, for example, a fossil organism where you don't have the sheath, again, make sure you use the lengths, include the second moment of area if you can, and exclude the extreme stuff, and you can get it down to about 89% or excuse me, 79% classification, correctly classified. The best case scenario is having both structures together using the lengths. You can get that down to around 10% or 90% classified correctly. And that's not even including the posterior probabilities, right? Those second most probable classifications. That's just the first most probable classifications. And what's really interesting that I want to point out here is it doesn't matter if you use the cross-sectional shape or not if you have both the sheath and the bone. Both come out 10%. So you don't need the cross-sectional shape if you have both the ungule and the sheath. You can get the same results, the same confidence out. So sheaths perform better than ungules, just slightly, but both gives the best results. Okay, this is just the LDA showing the classification. You can see how they're separating in space, so that's all really good. And then just showing that this is 10% with or without the second moment of area metric. Again, with the posterior probabilities, if you do happen to consider the second most probable, you can get in the neighborhood of 97% classified correctly, again, with or without the cross-sectional shape area. So all good news, right? I'm just going to skip over some of these conclusions because I went over them in the thing already, so that's all good there. The one thing I do want to point out is if you are reconstructing the sheath or trying to infer sheath shape from a fossil that doesn't have a sheath, you do need to be careful at how you're reconstructing that, because not everything is closely correlated, right? Those R-squared numbers weren't as great for some metrics as they were for others. So you need to take care in how you're doing that. And both the sheath and the ungule are contributing mechanically to the function of the claw. So using both structures is great. And the cross-sectional area only minimally improves the results. You don't really need it if you have both the sheath and the ungule to work from. And that brings us to Archaeopteryx. Is it climbing, perching, arboreal versus cursorial? What is it doing? All right, we want to resolve that confusion, predict the claw function using the information that we've learned from parts 1 and 2, and see how that it can be incorporated into the origin of flight, the ecological context for that evolution of that feature, and how that is reflected in the morphology of the claws. So basically, we're asking the question, is Archaeopteryx a roadrunner or is it a woodpecker? What is it doing? These are the two specimens of Archaeopteryx that I use. There's something like 17, 18 or so specimens. These are the two I used because they have claw ungules and sheaths preserved. And so I was able to take images of these specimens and measure both the ungule and the sheath on there. And because I have both the ungule and the sheath, I don't need to worry about the cross-sectional shape in these specimens because I can get a good, accurate prediction. I did also throw in, here's our friends, the downy woodpecker and the gray squirrel. I took a full set of claws from each of these animals and threw them into the analysis as unknowns just to test how the analysis predicted their function. I already know that they're scansorial, one's a mammal and one's a bird, but I wanted to test how the program was using that and compare that to how it classified Archaeopteryx, just to see if everything was functioning correctly. And here's what it looks like. I won't talk about the PCA so much or the LDA so much, but what you can see is that Archaeopteryx in yellow is grouping close to the downy woodpecker, which is Picoides. And in the LDA, it's also plotting close to the Sciurus, which is the squirrel. So it's lining up with scansorial forms, and those scansorial forms are lining up with the other scansorial and grouping together. So this is graphically what that looks like. Here are the posterior probabilities for that, and you'll notice that both the squirrel and the woodpecker, 99%, 95% right down the line, they are being classified as scansorial without hardly any doubt there. What about Archaeopteryx? Where does Archaeopteryx fall? Archaeopteryx, just as Archaeopteryx does, is generalist and weird and interesting. It is generalist, scansorial, and amplectorial. But I think what is most interesting is that it's nothing of anything else. You look at all the zeros: 0 for cursorial, 0 for digging, 0 for tensasorial. It's all scansorial, amplectorial, or generalist. So climbing and perching is playing an important role for Archaeopteryx in one way or another. At least that's what the claws say. Now, there could be many benefits to climbing and perching. You can have conservation of energy. It's been shown that it's more energy efficient to climb up a trunk and glide down to the next trunk. So woodpeckers do this as they forage for food through the woods. Also, predator avoidance. You can climb up and access high safe zones or access elevated perches for gliding and going to the next tree. Also, if you can climb up and glide from tree to tree, you don't have to run along the ground. If there are predators on the ground that would like to eat you, you can avoid those predators altogether simply by climbing and gliding through the trees. So lots of benefits to this behavior that would induce the evolution of such behavior, climbing and gliding. But what does that mean for Archaeopteryx and its evolution? Well, we like to refer to Archaeopteryx as what we call a morphological mosaic. What that means is that different features of its body plan are being used and exhibiting different functions. What this means is that if you take all of the aspects of Archaeopteryx, if you were to measure the wings and the claws and the limb bones and the foot and the skull, if you were to measure all that and throw all of that into analysis, Archaeopteryx basically comes up as a big mess, a big generalist mess, because it's combining all of these specialist functions, this mosaic of functions, into one whole. And you can't think about Archaeopteryx or any transitional fossil that way. Transitional fossils are often morphological mosaics because as they're going through that transition, in this case from terrestrial to flight, different body parts are adapting to different functions at different times in that evolution, in that transition. So there's not really anything we can look at today that does that. It's hard to find homologs or analogs for these things in modern organisms. What that also means is that you can't consider Archaeopteryx or any transitional fossil as a whole animal. You have to pick it apart and consider each structure separately. Each structure has to be examined and understood independently for it to make sense. In this case, the claws are saying climbing and generalist. There are some studies that have looked at the hind limb bones in Archaeopteryx, and they say cursorial. The hind limb bones of Archaeopteryx say cursorial. The claws say climbing. It's probably both. It's probably doing both. The claw and the limb morphology are highlighting different aspects of the overall life habit. Archaeopteryx is a really special, unique animal in that sense. And it just highlights the fact that we have to consider function first, then ecology. You can't just assume that it's doing something because the claw says so or because the hind limb says so. You really have to consider each character independently, the function of each character, and then think about the ecology that it is suggesting. We can look to baby birds today for limb coordination. What this means is that the baby birds today are using their arms in different ways than they're using their legs. So it's kind of like mosaic evolution. It's kind of like a transitional fossil. Once these birds grow up and become adults, they become very specialized for one thing throughout the whole body. But as young individuals, some of them display these different features similar to transitional fossils. And employing the wings and legs cooperatively offsets the detrimental effects of each one. So having an undeveloped wing means you can't really fly very well. Having a leg that doesn't run very well means you can't run very well. But using both structures in cooperation means that you can do all those things equally well, and that gives you a leg up, as it were. It gives you an evolutionary advantage, especially when there is nothing else in your environment that is doing that thing. So Archaeopteryx is one of these transitional forms that doesn't do anything really well, but it is specializing different structures for different functions so that it can have a benefit, that selective advantage by using these different limbs and these different body structures cooperatively. So essentially, the bird, in this case Archaeopteryx, can effectively engage in locomotion and behaviors that it wouldn't be able to do otherwise if it was relying on one structure or the other. And then I want to mention these interesting animals here because we see this happening in other transitions as well. If we look at the bats, a new hypothesis was just proposed called the interdigital webbing hypothesis, but it's proposing that in these fossil bats, they have claws on all the digits of the hand. Modern bats only have digits on the wing claw, the thumb claw. So what this means is that these bats were probably still climbing in the trees as they were developing powered flight. And only after they developed really good specialized flight did they lose the claws on the digits. We see that in the transition from birds. Archaeopteryx and the first birds have claws on the digits. They're probably climbing around in the trees until they get really good at flying, and then they don't need the digits, the claw digits anymore. I mentioned Ambopteryx, which also not only has claws on the digits, but it has wing membranes as well, right at the base of the birds there. And then was mentioned earlier, right? Randy Ermis's group just published on this lagerpetid fossil pterosaur relationship. And in that paper, in the Nature paper, there is in there mention about the claws of Dromomeron, which is in the neighborhood of 150 degrees for the ventral curvature. That's really, really high. That is in the Scansorial to Tensasorial range. So this is a repeated thing that we're seeing, right? Small-bodied animals, claws on the digits, climbing around in the trees. Once the transition to powered flight is complete, they lose the claws on the digits. And I think this is something that we see in Archaeopteryx and the birds. We're seeing it now in the bats, and it's popping up now in the pterosaur evolution as well. This is going to be a recurring theme that we're going to see in the transition of these evolutionary animals. All right, so that's the conclusions. Archaeopteryx is probably generalistic, splitting its time between the ground and the trees. Definitely, as far as the claws are concerned, climbing and grasping and perching in the trees was an important part of its lifestyle that needs to be considered. I already mentioned a lot of the benefits that these would have and the evolution of those things. Different body parts are reflecting different functions of the animal. And supports the hypothesis, and like I mentioned, of cooperative use of the forelimb and hind limbs. And like I just mentioned, I think it's a common theme in the transition to flight in all of these vertebrate groups. I want to quick run through some of these arm wavy things. So look at all of these projects and papers I haven't started. Oh, wow. One is, at some point, I am going to write a review paper describing in detail all of these functions, because there are more functions that I've identified than that I put into this analysis. One of those functions is grooming. Beavers have a grooming claw that they use to groom their fur. It's a specialized claw on the hind foot that they use for that function. There are certain families of birds that have what's called a pectinate claw, or a comb structure on the sheath. It's still uncertain what that's used for, although grooming seems to be the consensus for that, but that's an interesting thing. And then one of the most interesting things I find is in the pond turtle. These are where the pond turtles use their claws during mating behaviors. The only function for these claws is during a mating ritual, and they are commonly called slapping turtles, where they slap or touch or wiggle around the face of the female during the mating ritual that you can see here. And that's all they use their claws for. They don't dig, they don't grab things with them or anything. It's only in the males. It's hormonally induced during maturation. This is a very specific, interesting, unique claw function. Sexually selected mating behavior here. And then lastly, I do want to mention about Utahraptor and the dromaeosaurid dinosaurs. They're often depicted as having these nice sharp claws. Definitely the digit 2 claw was sharp, but it's the other claws that I'm interested in. If we look at modern animals like the ostrich, large-bodied cursorial animals, they have the hoof-like flattened claws on them. So I think we need to reconstruct the claw sheaths, at least on the digits 3 and 4 of the raptors, as blunted hoof-like claws. That would make the most sense for what they're using their claws for, particularly large-bodied raptors that are probably running cursorial very quickly. They would have blunted claws. As far as the digit 2 claw is concerned, there's a Seriema bird that has this interesting condition. You can see the foot there. This is the sheath reconstructed on the digit 2 claw of Utahraptor, compared to the ungule there for a Seriema. Similar shape, and so this is the reconstruction that I would put on the sheath of the Utahraptor claw. And then lastly, I think there's a very interesting model for why Utahraptor and other dromaeosaurid dinosaurs evolved that second digit claw specialized on their foot. And it has to do with the cheetahs. Cheetahs are one of the fastest running quadrupeds on land today. They achieve those speeds and that agility through their claws. They have less curved. They're not like other cat claws. They're very specialized, where they are less curved, they're a little bit blunted, and they function essentially as cleats to give the animal traction and friction for high-speed chases. However, once the animal catches up to a prey item, they use a extremely hooked and enlarged digit 1 dew claw to hook the animal and bring it down. So some of the claws are being used for cursorial. There's a specialized claw that is used gripporial or for grappling, a tensasorial function to bring that animal down. I suspect that the Utah raptor and the dromaeosaur dinosaurs specialized the inner claw for grappling, like we see in the peregrine falcon that I showed earlier, for gripping and grabbing down prey, and that they use their other claws for running fast. And that this was a claw specialization transition in their evolution, and that we can look to the cheetah to kind of see a similar thing happening there. So thanks for attending or watching. I'm not sure which this involves, but thanks for doing it, and I'll take any questions. I'll sign out here and we can look at questions.
Hi, does anybody have any questions? I don't see any in the chat yet. Put it in, or if you'd like at this point, you could unmute and ask directly. Tracy, your talk was very well detailed, so I have a feeling you have probably explained what we wanted to know.
Tracy,
there's a lot of stuff that I didn't talk about too. Yeah,
did you do comparisons of the claws across the feet of these animals? You don't say in your analyses. You don't say 1, 2, 3, and 4. You just here's a claw. You know, this animal.
It's I used one claw per animal essentially, and it was the claw that I used. I assigned it the function that it did. So there are animals that specialize their claws, and there are animals that use the front claws different from the hind claws. And but in the specimens and the time that I had to put it together, I just picked one claw per animal. And that's why I incorporated the squirrel and the woodpecker at the end to look at interdigital variation. Those were a complete set of claws from each of those animals combined with sets from Archaeopteryx just to see what the variation would do. And it wasn't too bad. They clustered together pretty well and gave some good results.
Tracy, it's Ron.
Yeah.
Got a question on the suspensorial claws. You mentioned the sloths were the only one that you had looked at there. What about an animal such as bats hanging from a ceiling?
Those ones are functioning more like a scansorial type of function. So where you have a vertical substrate and the claw is attaching to that substrate to hang, that's a scansorial type function. So that's for climbing and for grabbing up like that. Suspensorial is where you have a beam and the claw is hooked over the beam.
Okay.
Some bats might do that, on small branches, but they're such small animals that I think for the most part they're doing this type of activity with the claws. They're more gripping the substrate in a scansorial fashion than hooking around or grasping a branch or something. Yeah.
Okay, thanks.
Yeah.
Anyone else? Just feel free to unmute and hop in.
I've got one, Tracy. This is
Yes.
Josh.
Hey, Josh.
Jim took one of my questions that I had about variation across digits within the same animal. But another question I have is about a different group of critters. With lizards, you get quite a bit of variation in ecology. So I'm just curious if you've dug into the literature at all to see if there's any any information on ungule versus sheath variation in lizards related to ecology, whether it's some of the more arboreal lizards. Yeah, I think about things like Scalporial, where you have some Scalporial that are very much tree dwelling, or at least can be, like Scalporial clarki down in Arizona. Did you chase anything with lizards there?
Yeah, there actually is quite a bit of literature focusing on lizards and substrate use. Specifically, they use anolis lizards, as well as trying to think of what the other one is. Oh shoot, it's the same one that the Komodo dragons belong to. Varanids, the varanid lizards. They are really diverse in what they do as a group. Some of them are arboreal, scansorial. Some dig in the sand, burrowing, and there's aquatic ones and all sorts of stuff. So Yeah, there's been some work on that. The main flaw with a lot of those studies is the functional categories. They have not considered functional claws, and they're still categorizing them by habitat. You know, arboreal, aquatic, terrestrial, and that's not quite going to get you there. That being said, there is quite a bit of literature as far as looking at the relationships between the ungule and the sheath. That has also been attempted by a few studies, although they suffer from the same problems of the functional categories not being up to snuff, as it were. And I think that's where some of the conclusions are going a little bit off track in that, although there is some literature on that.
Gotcha. Thanks.
Yeah.
So we do have a couple of chat questions.
Cool.
Ashley Allred is asking, seeing how many different skills, subjects you use for your research, it made me wonder which school subject was your most favorite as a student, besides paleontology, of course.
Well, I'll give a cop-out answer. It was science. But it really was the science classes, you know, and it was the classes where I had some really good teachers that would allow me to kind of pursue my own questions in the chemistry lab or in a biology lab or something like that. And that's where I really kind of thrived and really kind of came into my own, is like trying to answer questions on my own, but also trying to come up with ways to answer that question. You know, I think problem solving classes, Gary and I, like Jim mentioned Gary, and I've talked with other people. One of my career goals is to go into education at a community college or something where I can talk to students about how to problem solve. You know, what does the scientific method do for you rather than just a body of knowledge, right? How does it make you interact with your world in a meaningful, constructive way? And that's, looking back on my schooling, like I said, the classes where I was able to kind of do that on my own, or where I was taught that, how to do that and taught that, those are where I really thrived. So I think you're right. You know, looking at multiple ways to approach a problem, and then just trying it out. You know, testing some things to see what works and what doesn't. That's kind of how I approach this. One thing I'll say about that is one of the reviews I got back on the first chapter, somebody called it a shotgun approach to the problem. And I took that as a compliment. That's what I intended it to be. Just throw everything at the problem and see what worked and what didn't, and take what works and use that, and discard what doesn't work. And I think that's how we progress. That's how we get through our lives, and that's how we should interact with everything. So, yeah.
Excellent. And then Calvin Black asks, have you studied any other dinosaur types, their claws? I'm particularly interested in where sauropods would fall out in the analysis, in this analysis.
I'm interested in that too. So Adam Marsh contacted me with the new Drepanosaurs that they're describing down there, and he did a functional PCA analysis on the claws, and came up with that they were Scansorial and some other things, I think. But he's wanting to throw those claws through my analysis and see if he can repeat the results or see if it shows something different. For sauropods, there's quite a bit of literature on them using their claws to scratch dig, or to scratch out nests to lay eggs, or to do different things like that. I haven't looked at those claws in any detail yet, but I am keeping a list of papers that describe function in fossil claws of all types and groups of animals. At some point, I may return to that and start throwing some things through the analysis once I get these other chapters and these other papers published and get the framework and the foundation in the literature. Then I could start addressing some of those things.
And then got another one from BJ Nichols, wondering if you've looked at ichthofossil claw impressions and what your new work might do to help tease out function or morphology.
Yeah, I've also been keeping a list of papers that describe more specifically Andromaeosaur tracks that preserve claw impressions, because I'm interested to see if you can pick out the bluntedness of those claws in the claw impressions. You know, if my hypothesis that those should be blunted and hoof-like in those cursorial animals, we should be able to see that in the tracks and the trackways. So that's another avenue that, yes, I've considered, and I've been, again, keeping a record of papers in the background. Again, look at all these things that I haven't done, right, that need to get done. At some point, the job will get done, and we'll look at those. But yeah, that's a great avenue to go look at it in.
Well, one
Yeah.
thing, just to throw something in on that,
Yeah,
dromaeosaurs, leg proportions are such that they've been pretty universally considered to not be runners because they have leg proportions that are not very efficient for running compared to, say, ornithomimids or even tyrannosaurs.
right.
But they have short feet and shorter tibias than femurs typically. But we're seeing that real difference, with the young Utahraptors, that the young ones look like they're runners, and the adults.
And you look at the stride lengths on some of the dromaeosaur tracks, at least the mid-sized bodied ones, and they're cruising pretty fast for their size with those stride lengths.
Yeah, you looked at the Mill Canyon stuff, I bet.
That stuff too, yeah.
Yeah, that's nice.
Yeah. So I think, cursorial, it's all kind of relative. I don't like the terrestrial category because, I'm terrestrial. So, but I don't have a cursorial body form, at least not anymore. Now that I get older, right? It doesn't have to. So, I think, again, it comes down to those categories. You got to classify things correctly. You got to have evidence for those classifications and use those things appropriately. But as far as dromaeosaurs, I think you're right. It's going to be interesting to look at those animals. And I think, again, with the cheetah example, looking at the, just how fast did they run? Just how specialized did they get? And what are they using that for? And where did it come from? What does the basal animal look like? And all of those great questions.
Well, the tail morphology, they have the balancing tail.
Right.
Use their tail, running.
Yep, exactly like that, just like the cheetahs. Yep. Yep. I think there's something there.
Yeah.
Yeah.
So does anybody else have any more questions for Tracy? Well, Tracy, I want to thank you very much. That was an excellent presentation, and I've already been comparing my dog's claws to my cat's claws as I was sitting here. I'm sure they were not happy about it, but anyway, it does become fascinating seeing the differences.
Yep.
So
Thank
it
you.
is,
Thanks for
as
inviting
you
me to speak. Yeah.
yeah. So everyone, do remember, if you want to hear any parts of this again, it will be eventually published on YouTube, our YouTube channel, by BJ, and we'll be back again in January after the new year with Joshua Lively will be presenting to us. And hey, everybody have a really good and safe holiday and new year. Please stay safe. We want everybody to, be with us for our next meeting. And thank you for coming. And thank you, Tracy.